Good evening, everyone. Myself Himanshu, and I welcome you to this online platform of Unacademy where we will be studying about CSIR UGC NET. So, I welcome everyone online, and please like, share, and subscribe our channel with the people who are preparing for this examination, and please share and like as much as possible. So, before we start, I would like to tell that you need to go to the Google Play Store where you need to install the Unacademy Learning app. And after installing the Unacademy Learning app, you need to select your goal, where you need to select a CSIR UGC NET category. And once you select the category, you will have access to various top educators, top educators free courses that are available on the Unacademy platform. And if you want to go for the plus platform, then you can. If you want to go for more, you need to go for the plus Unacademy Plus subscription. So you will be getting extra notes, you will be getting extra quiz material, you will be getting extra mock tests. So all these are the advantages of the plus course. And once you get the subscription in a particular category, you will have access to various educators in that particular category. So you need not go for separate subscription for that particular for those particular educators. So यहाँ पे CSIR UGC NET का subscription is available for 1 month, 3 months, 6 months, 12 months and 24 months. Well, I would recommend that each one of you should aim for 12 months to 24 months of subscription, and you can use my code HY10 for getting 10% off. So then you need to proceed to the payment, and payment can be done either by debit card or credit card or by EMI, and these EMI are all no cost EMI. Also, the payment has been done by net banking. So any of the mode, so the entire course, if you go for 12-month subscription, you will get for 10,000. If you apply my code, you will be getting it for 10,395 instead of 11,550. So let's begin today's session. And before that, I would like to tell to each one of you that we will be holding these live classes here on Unacademy's YouTube channel as well. So you can join these classes and attend these classes. So I welcome everyone. So in today's class, we will go for the brief about, we'll be doing the brief about the cell cycle and the mitosis and meiosis in general. So let's start today's class. So as you all know that eukaryotic chromosomes, they contain the DNA and the protein, and the chromosomes, they carry the genetic information. So eukaryotic chromosomes, they contain the DNA and the protein, and the chromosomes, they carry the genetic information. And this genetic information need to be passed from one stage to another stage, and that is where cell division comes into play, where it will be leading to the distribution of the genetic material into the progeny from the parent cell. So this is the chromosome, and in the condensed state, this is the structure. So these are the telomeres that are present at the end of the chromosome. This is the center region is known as the centromere, and this shows the structural evolution, where the chromosomes are condensed and then they are packed into histones, and which ultimately is converted into DNA. So having base pairs. So this is how the cellular organization works. So this one is a supercoil within the, this one is a DNA double helix, and this is packed using histone proteins. So histone proteins are present, and it will be forming a chromatin fiber which will undergo supercoiling within the chromosome. So this is how the DNA is packed in a cell. So when cell divides, the chromatin fibers are very highly folded, and and become visible in the light microscope as chromosomes. So when the cell divides, the chromatin fibers are very highly folded and become visible in the light microscope as chromosomes. And during the interphase, the chromatin is more extended, a form used for expression of genetic information. So interphase के समय chromatin is more extended into a form that is used for expression of genetic information. So when the cell undergoes cell division, the chromatin fibers are highly folded and they become visible in the light microscope as chromosomes. And during the interphase, that is between the divisions, the chromatin is more extended into a form that is used for expression of genetic information. So DNA is organized into informational units called genes. So DNA is organized into informational units called as genes, and chromosome contain 100 to thousands of genes. Chromosomes, it contains 100 to 1000 of genes. तो व्हाट इज सेल साइकिल? सो सेल साइकिल एक सीक्वेंस होता है ऑफ सेल ग्रोथ एंड डिवीजन. So cell cycle is a sequence of cell growth and division, and cell cycle is the period from beginning of one division to the beginning of the next. So cell cycle is a sequence of cell growth and division, and cell cycle is the period from the beginning of one division to the beginning of the next. Good evening everyone. Sayo Ram, good evening. और जितना टाइम लगता है इसको कंप्लीट करने में वन सेल साइकिल को, इट इज़ नोन एज द जनरेशन टाइम. तो हम लोग ने कल माइक्रोबियल फिजियोलॉजी के लेक्चर में देखा था व्हाट इज़ जनरेशन टाइम. तो this is all about generation time. The time it takes to complete one cell cycle is called the generation time. And cells, they divide when they reach a certain size, and cell divisions involve mitosis and cytokinesis. And mitosis involves the division of chromosomes, and cytokinesis involves the division of the cytoplasm. And mitosis without cytokinesis results in multinucleate cell. So this is a eukaryotic, general eukaryotic cell, where beginning of one division to the beginning of next takes place. There are various stages in eukaryotic cell cycle. First there is interphase, then there is a first gap phase, then there is synthesis phase, then there is second gap phase, then there is M phase, which consists of mitosis and cytokinesis. तो ये एक जनरल ओवरव्यू है सेल साइकिल का. पहले जीवन फेज होता है, देन देर इज एस फेज. जीवन फेज इज नोन एज द ग्रोथ फेज, इसको ग्रोथ फेज भी कहते हैं. देन देर इज एस फेज जहाँ पे डीएनए का रेप्लिकेशन होता है. एंड देन देर इज G2 स्टेज where DNA supercoils and it condenses and G2 से फिर एक M phase आता है and M phase is the mitosis phase. और M phase में anaphase, metaphase, prophase, metaphase, anaphase, telophase is present, and then cytokinesis takes place before the cell goes into again G1 phase. And this one is the interphase. Interphase consists of G1 phase, S phase and G2 phase. So chromosomes, they become duplicated during interphase, and cells are very active during interphase, synthesizing biological molecules and growing the G1 phase. And S phase is marked by DNA replication, and G2 phase occurs between the S phase and the mitosis. Despite there are several differences between the prokaryotes and the eukaryote, there are several common features in their cell division processes. So replication of DNA must occur, and segregation of original and its replica will follow. And cytokinesis is end the cell division process. So डिस्पाइट सेवरल डिफरेंसेस, बहुत सारी सिमिलैरिटीज़ भी हैं, कॉमन फीचर्स भी हैं बिटवीन प्रोकैरियोट्स एंड यूकैरियोट्स. जैसे कि रेप्लिकेशन ऑफ डीएनए मस्ट अकर, देन देयर इज सेग्रीगेशन ऑफ ओरिजिनल एंड इट्स रेप्लिका इट फॉलोज, एंड साइटोकाइनेसिस इट एंड्स इन द सेल डिवीजन प्रोसेस. So despite whether the cell was eukaryotic or prokaryotic, these basic events they must occur before any cell division takes place. So in this case, the hereditary material is passed on to new cells by mitosis or meiosis, and it helps in the, and then there is cell division and growth and reproduction. Then there is interphase, then there is mitosis, then there is cytokinesis, and then there is meiosis. तो सेल डिवीजन के समय क्या होता है, देयर इज क्रोमोसोमल पैकिंग ऑफ डीएनए. there is chromosomal packing of DNA that allows efficient distribution of genetic material during cell division. And life cycle requires two distinct types of cell division processes. Number one is mitosis, number two is meiosis. And in cell division, one cell becomes two cell during an organism's life cycle. So mitosis is the nuclear division plus cytokinesis, and it produces two identical daughter cell following following these following steps. Uh, then there is prophase, metaphase, anaphase, and telophase. And interphase is often included in the discussion of mitosis, but interphase is technically not a part of mitosis, but it rather encompasses stages of G1S and G2 of the cell cycle. So, so the interphase, it consists of the G1 phase, S phase, and G2 phase. And G1 phase is the growth phase where the cell grows and it grows in size. And then there is S phase where DNA is replicated. DNA का replication होता है S phase में. एंड देन देर इज़ G2 फेज जहाँ पे डीएनए का सुपर कॉलिंग कंडेंस होता है और मशीनरी माइटोसिस की असेंबल होती है. उसके बाद माइटोसिस का फेज़ आता है जिसे एम फेज़ भी कहते हैं. उसमें अलग अलग स्टेजेस हैं जैसे कि प्रोफेज, मेटाफेज, एनाफेज एंड टीलोफेज. एंड लास्ट में आता है साइटोकाइनेसिस वेयर सेल इज डिवाइडेड इनटू टू इक्वल सेल्स with equal genetic material. तो अब हम लोग इंटरफेस के बारे में स्टडी करते हैं. The cell, it is engaged in metabolic activity and performing it's prepared for mitosis. Chromosomes are not clearly discerned in the nucleus, although a dark spot called nucleolus may be visible. The cell may contain a pair of centrioles, both of which are organizational sites for microtubule. So the cell is engaged in metabolic activity and performing its prepare for mitosis, and chromosomes are not clearly discerned in the nucleus, although a dark spot called the nucleolus may be visible. The cell may contain a pair of centrioles, both of which are organizational sites for microtubules. Then there is prophase. In the prophase, chromatin in the nucleus begins to condense and becomes visible in the light microscope as chromosomes. The nucleolus disappears. Centrioles, they begin moving to opposite ends of the cell, and fibers extend from the centromeres. Some fibers, they cross the cell to form the mitotic spindle. तो जैसा कि आप यहाँ पे देख रहे हैं, the centrioles, they start moving towards the end of the cell. So इनका movement हो रहा है and it will come to this place, somewhere at this place. And it will assemble over here. Nucleolus, it disappears, and chromatin, it becomes to condense and become visible in the light microscope as chromosomes. So chromosomes का occurrence यहाँ पे होना start हो जाता है. So chromosomes are visible during the prophase, and nucleolus disappears, and centrioles begin moving to opposite ends of the cell, and fibers extend from the centromeres. And some fibers, they cross the cell to form mitotic spindle. So some fiber, they cross the cell to form mitotic spindle. Then there is pro-metaphase. In the pro-metaphase, the nuclear membrane dissolves, marking the beginning of pro-metaphase. Proteins attach to the centromeres, creating the kinetochores, and microtubules attached as the kinetochores and the chromosomes begin moving. Microtubules attach at the kinetochore and the chromosomes begin moving. Next comes the metaphase, where the spindle fiber line the chromosomes along the middle of the cell nucleus. This line is referred to as the metaphase plate. Polar microtubule extend from the pole to the equator, and typically they overlap. Kinetochore microtubules extend from pole to the kinetochore. This organization helps to ensure that in the next phase, when the cell's chromosomes are separated, each new nucleus will receive one copy of each chromosome. This organization, it helps to ensure that in the next phase, when the chromosomes are separated, each new nucleus will receive one copy of each chromosome. This one is the general assembly during the metaphase. So आप देख रहे हैं कि यहाँ पे metaphase में the polar microtubules, they extend from pole to the equator, and the kinetochore microtubule extend from poles to the kinetochores. So this ensures that all the chromosomes, they are separated, and they all lie in the middle of the cell nucleus. This line is referred to as metaphasic plate. So metaphasic plate comes into existence. Then there is anaphase. In the anaphase, the paired chromosomes, they separate at the kinetochores and move to the opposite sides of the cell. So the chromosomes are pulled by the kinetochore microtubules to the poles and form a V shape. Motion results from a combination of kinetochore movement along the spindle microtubules and through the physical interaction of polar microtubules. So this is the anaphase, where the paired chromosomes, they separate at the kinetochores and they start moving towards the opposite side of the cell. So in this case, the chromosomes, they separate at the kinetochore and they move to the opposite side of the cell. And chromosomes, they are pulled by the kinetochore microtubules to the pole and form the V shape. So this is how the chromosomes are pulled. and motion results from a combination of kinetochore movement along the spindle microtubule and through the physical interaction of polar microtubules. So this is all what is happening in the anaphase. Then उसके बाद आता है telophase, जहाँ पे chromatids they arrive at the opposite poles of the cell. Chromatids they arrive at the opposite poles of the cell and a new membrane form around the daughter nuclei. The chromosomes disperse and are no longer visible under the light microscope, and spindle fiber disperse and cytokinesis will start. So this is the telophase, जहाँ पे chromatids arrive हो जाते हैं opposite pole पे and new membrane are found around the daughter nuclei, and the chromosomes disperse and are no longer visible in the light microscope, and spindle fiber disperse and cytokinesis will start. So this is the cytokinesis. So in the animal cell, cytokinesis results when a fiber ring composed of a protein called actin forms around the center of the cell, and it contracts, pinching the cell into two daughter cells, each with one nucleus. In a plant cell, synthesis of new cell wall between two daughter cells rather than the cleavage furrow in the cytoplasm. So this is the cytokinesis. So cytokinesis has occurred and two new cells are formed. This one is cell 1, this one is cell 2. So two new cells are formed. So this is the interphase or the preparatory phase. Next is the prophase. This one is the pro-metaphase. This one is the metaphase. This one is the anaphase. You can see the chromosomes, they are attached to the kinetochore and they are moving and they are forming a V shape. They start their movement. Towards the pole. Then there is telophase, and then there is cytokinesis, where cells have been divided into two equal cells. So there is interphase, जहाँ पे cell is carrying out the normal cell activities and chromosomes become duplicated. Then there is early prophase, where the nuclear envelope begins to disappear and nucleolus also disappears and long fibers of chromatin become evident and begin to condense as visible chromosomes. Then there is late prophase, where chromosomes continue to shorten and thicken, and spindle forms between centrioles which have moved to the poles of the cell, and kinetochores begin attaching to the microtubules. Then there is metaphase, where the spindle fibers attach to the kinetochores of the chromosome which line up the cell's midplane. Then there is anaphase, where the chromatids separate at the centromeres and one group of chromosomes move towards the each pole. Then there is telophase, where chromosomes have arrived at the poles and the nuclear envelope begins to form. And at the end there is cytokinesis, which produces two daughter cell. So interphase is the preparatory phase, where daughter cells formed are genetically identical to the parent cell, and once again interphase starts for further cell division into next cell. So this is how the things go on in an animal cell, and this one is in the case of the plant cell. So in the case of plant, there is a formation of a cell plate. Cell plate formation occurs where there is no cell plate formation that is occurring in the animal cells. This one is the metaphase. As you can see, the chromosomes, they lie at the equatorial plane. This one is the metaphase. This one is the anaphase, where the V-shaped chromosomes are seen. Then there is the telophase, where the chromosomes have arrived at the poles and the nuclear envelope begins to form, and cytokinesis produces two daughter cells. So this is how the cell division takes place. तो अगर आप लोग के पास कोई डाउट्स हैं, कोई भी क्वेश्चन है, तो यू कैन पोस्ट इन द कमेंट बॉक्स. तो ये ड्यूरिंग द इंटरफेस है, एंड डीएनए रेप्लिकेट्स ड्यूरिंग द एस फेज, जहाँ पे पेयर ऑफ सेंट्रियोल्स होंगे, एंड क्रोमैटिन is not that condensed. Then chromatin starts condensing and prophase is formed, and then metaphase is formed, then anaphase is formed, and then telophase is formed, where the cell telophase and cytokinesis takes place together, and there is division of the cytoplasm. So the general reproduction, it is in two, occurring in two forms. One is the asexual reproduction, other one is the sexual reproduction. So there are two modes of reproduction. One is the asexual mode, other one is the sexual mode. So during asexual reproduction, a form of duplication using only mitosis. For example, a new plant grows out of the root or a shoot from an existing plant, and it produces only genetically identical offspring, since all divisions are by mitosis. So this is the case of asexual reproduction, where it is a form of duplication using only mitosis. And a new plant, example, a new plant that grows out of the root or a shoot from an existing plant, and इसका ये होता है कि it produces only genetically identical offspring, since all divisions are by the mitosis. So in asexual reproduction, all the cell divisions, they occur by mitosis. So in mitosis, one parent cell, it divides into two daughter cells. So simple division of two daughter cells will take place. So this is the case of asexual reproduction. Next, there is a case of sexual reproduction, where formation of new individuals occur by combination of two haploid sex cells. By combination of two haploid sex cells. So new individuals are formed by combination of two haploid sex cells or the sex gametes, So one is from the male progeny, other one is from the female progeny. So two haploids they meet, forming a diploid condition. So fertilization is the combination of genetic information from two separate cells that have one half the original genetic information, and gamete for fertilization usually come from separate parents. Gamete for fertilization usually come from separate parents. So female produces an egg and male produces sperm. So both gametes are haploid with a single set of chromosomes. The new individual is called a zygote with two sets of chromosomes that are diploid, and meiosis is a process to convert a diploid cell to a haploid gamete and cause a change in the genetic information to increase the diversity in the offspring. Then there are chromosomes in a diploid cell. This is a summary of chromosome characteristics. So there is a diploid set for humans where 2n is equals to 46, and then there are autosomes which are homologous chromosomes, one from each parent. There is 22 sets of 2. Then there is one sex chromosome that have one set. Then there is female sex chromosomes that are homologous. Then there are male sex chromosomes that are non-homologous. So number of sets of chromosome in a cell. There is haploid, that is one set of chromosome. Then there is diploid, there are two sets of chromosome. Most plants and animal adults are diploid, and egg and sperm are haploid. So most cells in the human body that are they are produced by mitosis. These are the somatic cell lines, and cells that become gamete are referred to as germ-line cells. There are vast majority of cell divisions in the human body that are mitotic, with meiosis being restricted to the gonads. So what is a diploid cell? So it is having a characteristic number of chromosome pair per cell. They are the homologous chromosomes, which are similar in length, shape and other features and carry similar attributes. Then there are haploid cells which contain only one member of each homologous chromosome pair. So the general growth that occurs is the case of classical case of mitosis, where the genetic information that remains the same. So it is 2n condition, and over here also it grows into a 2n condition, while meiosis occurs in the gametes, in the gonads, and 1n from each male progeny and 1n from female progeny, and then there is a fusion formation of zygote. So this occurs in the 2n form, and by mitosis it leads to the formation of the new individual. This is how the life cycle of human takes place. So in meiosis, the diploid cells undergo meiosis to form haploid cells. So diploid cells undergo meiosis to form haploid cells, and meiosis potentially produces 4 haploid cells. Meiosis, it involves two separate divisions. So two successive nuclear divisions occur. One is meiosis 1, that is the reduction division. Other one is meiosis 2, that is the equational division. So meiosis 1 reduces the ploidy level from 2n to small n, that is the reduction phase, and while meiosis 2 divides the remaining set of chromosomes in a mitosis like process, that is the division. So in meiosis, homologous chromosomes are separated into different daughter cell. So meiosis 1 and meiosis 2 each include prophase, metaphase, anaphase, and telophase. This is the first division of meiosis 1, where prophase 1 is the most important stage of the meiosis. During this stage, many crucial events occur. So in prophase 1, the spindle appears, the nuclear envelope it disappears, and the DNA of the chromosome begins to twist and condense, making the DNA visible to the microscope, and each chromosome actively seeks out its homologous pair, which also has a sister chromatid. The two replicated homologous pairs find each other and form a synapse. The structure formed is referred to as tetrad, and the point at which two non-sister chromatids intertwine is called a chiasma. Sometimes a process known as crossing over occurs at this point. So this is where two non-sister chromatids they exchange the genetic material, and this exchange does not become evident, however, until the two homologous pairs separate. So this is the prophase 1 of meiosis. So this is the chiasma formation where segments of DNA are exchanged at the crossover point. So this is the chiasma formation that takes place, and this is how the spindle fiber it forms. This is at the beginning of the anaphase 1, and during prophase 1 there are segments of exchange DNA that are exchanged at the crossover levels. So prophase 1 it includes synapses and crossing over. Homologous chromosomes pair and undergo synapsis. One member of a pair is the maternal homologue, while the other is the paternal homologue, and synapsis is the association of four chromatids, two from each homologue. So synapsis is the association of four chromatids, two from each homologue. So one member of a pair is the maternal homologue, and other one is the paternal homologue. And homologous chromosomes they pair and they undergo synapsis. So in metaphase 1, the tetrad they line up at the equator, along the equator, and anaphase 1 results in the separation of homologous pairs. Cells are haploid at this point. Telophase 1 results in a brief reappearance of nuclear envelopes and the spindle disappears. The cell waits momentarily during the interkinesis, and the interkinesis separates meiosis 1 and meiosis 2, and no DNA synthesis occurs. So this is the second division, that is the meiosis 2. So in prophase 2, the spindle reappears and the nuclear membrane fragments. In metaphase 2, the chromosomes they align at the equator. In anaphase 2, the sister chromatins they separates. And in the telophase 2, the nuclear envelope reappear and the 4 haploid cells are the result. So this is a difference between meiosis 1 and meiosis 2. So this is meiosis 1. So this stage is meiosis 1, and this stage is meiosis 2. तो यहाँ पे ये स्ट्रक्चर है ड्यूरिंग द इंटरफेज के समय का ये स्ट्रक्चर है. तो ये दिस इज द क्रोमोसोम स्ट्रक्चर. यहाँ पे सेंट्रोमियर होता है बीच में और क्रोमेटिड फॉर्म में होते हैं. बट एट द स्टेज ऑफ प्रोफेज 1, दिस दे the sister chromatids they separate. Then there is metaphase 1 where the chromosomes they lie at the equatorial plane and the spindle fiber they attached at the to the centriole. Then there is anaphase 1 where they separate and move towards the fold. Then there is a telophase 1 जहाँ पे two different uh, cells are starting to form, and then there is prophase 2. Similarly, there is metaphase 2, anaphase 2, and telophase 2. So germline it undergoes gametogenesis. So germline में it undergoes gametogenesis. So spermatogenesis produces the sperm and oogenesis typically produces egg or a single ovum or two or more polar bodies. So this is spermatogenesis. This is oogenesis. सबसे पहले there is mitosis and spermatogonium is formed or the oogonium is formed, and then there is first meiotic division leading to the formation of uh, primary oocyte and the secondary oocyte. So the secondary oocyte is all occurring in the haploid stage. It undergoes second meiotic division to form the spermatids or the ootid. Then there is differentiation and maturation to form the sperm cells and the ovum or the egg cell. So it is all in the end state or the haploid state. So before you Before we finish today, I would like to again remind you all that आप लोग सब लोग गूगल प्ले स्टोर पे जाएं और वहाँ पे अनअकैडमी का लर्निंग ऐप है उसे इंस्टॉल करें. And then you need to select your goal before you do the login. And select your goal category, you need to select CSIR UGC NET. So once you do that, you will get access to various top educator courses in this particular category. And if you want to go for more, you need to go for the plus subscription. So the plus subscription is available for 1 month, 3 months, 6 months, 12 months and 24 months. So I would recommend that you go for the 12 months and 24 months of CSIR UGC NET subscription, and you can always use my code HY10 where you will be getting 10% off. So you can confirm the details and you can proceed to the payment. Now the payment can be done either by credit card or debit card or by the EMIs, and these EMI are the no cost EMI, or you can do the payment by the net banking. So the entire course will be available for 10,395 instead of 11,550. So this is all for today. So if you have any doubt, any queries, you can post. So thank you everyone. So if you have any doubt or any query, then you can post in the comment box. अगर आप लोग के कोई भी क्वेश्चन है तो you can ask them by pasting it in the comment box. And please like and share and subscribe to this channel where you will be please like and share and subscribe to this channel with as much as possible with the people who are preparing for this examination. And please don't forget to use my referral code HY10 for getting 10% off on your next Unacademy subscription. अगर आप लोग के कोई भी सवाल हैं तो उसे आप लोग कमेंट बॉक्स में टाइप कर सकते हैं. आई कैन सी योर कमेंट्स. और आप लोग से मैं सजेशन भी चाहूँगा कि आप लोग ये बताएं कि हम लोग अब what more you are expecting that we can take in the upcoming lectures. तो आज हम लोग का अगर हम लोग देखें तो हम लोग सिलेबस के फर्स्ट एंड पे आ गए हैं. हम लोग वी हैव फिनिश्ड विद द वी हैव फिनिश्ड विद द माइक्रोबियल फिजियोलॉजी. हम लोग ने माइक्रोबियल फिजियोलॉजी स्टार्ट करी थी. हम लोग ने ओपरॉन अच्छे से कर लिया था. वी हैव स्टडीड अबाउट द ऑर्गेनाइजेशन ऑफ जीन्स एंड क्रोमोसोम्स. हम लोग ने स्ट्रक्चर एंड ऑर्गेनाइजेशन ऑफ तो हम लोगों ने स्ट्रक्चर एंड ऑर्गेनाइजेशन ऑफ फंक्शन ऑफ इंटरसेलुलर ऑर्गेनेलिज हम लोग ने देख लिया था. हम लोग ने मेम्ब्रेन स्ट्रक्चर एंड फंक्शन देख लिया था. हम लोग ने आज सेल डिवीजन एंड सेल साइकिल स्टार्ट किया है. एंड इस दिस अपकमिंग वीक में हम लोग माइक्रोबियल फिजियोलॉजी और सेल डिवीजन एंड सेल साइकिल को समराइज करेंगे. इससे पहले हम लोग ने मॉलिक्यूल्स एंड देयर इंट्रैक्शन रेलेवेंट टू बायोलॉजी वाला सेक्शन किया था, जिसमें हम लोगों ने ये सारे सेगमेंट कवर किए थे। नेक्स्ट वी मूव टू द बायोएनर्जेटिक्स, जहाँ पे हम लोग ने ग्लाइकोलिसिस, ऑक्सीडेटिव फॉस्फोरिलेशन एंड ये सब कवर किया था। देन वी केम टू द एंजाइम्स एंड और द एंजाइम काइनेटिक्स। Then we came towards the conformation of proteins, which included the Ramachandran plot, secondary structure, domains, motif, and fold. We also studied about the stability of proteins and nucleic acid, and in the end, we studied about metabolism of carbohydrates, lipids. तो ये वाला पार्ट थोड़ा सा रह गया, बट मेजॉरिटी ऑफ द सिलेबस हमारा जो पार्ट 1 का मॉड्यूल था, मॉलिक्यूल्स एंड देयर इंटरेक्शन रेलेवेंट टू द बायोलॉजी, उसका हो गया है। एंड मॉड्यूल 2 जो है सेलुलर ऑर्गेनाइजेशन का, इसका हो गया है। तो आप लोग बताइए नेक्स्ट कौन सा मॉड्यूल हम लोग को स्टार्ट करना चाहिए। यू कैन पोस्ट योर कमेंट्स इन द कमेंट बॉक्स। आप लोग और क्या चाह रहे हैं, और हम लोग की एमसीक्यू की सीरीज़ चल रही है, जिसको हम लोग डेली मॉर्निंग में, आफ्टरनून, डेली सॉरी डेली हम लोग 12 PM पे हम लोग करते हैं, सुबह के समय, जहाँ पे हम लोग इम्पॉर्टेंट एमसीक्यू डिस्कस करते हैं। एंड शाम को हम लोग की 7 PM पे क्लास होती है, जहाँ पे हम लोग कंसेप्ट को देखते हैं, थ्योरी समझते हैं, चीज़ों की थ्योरी समझते हैं। तो what more should we start? You please comment in the comment box. So thank you guys, thank you everyone. Now I am ending this session.